In this video, we're going to be talking about hormonal signaling. In other words, how do you get a response in a target cell? Remember that what makes a target cell is that it has the right receptor, the receptor that fits the hormone. Whether or not a target cell actually responds depends on a few different factors. First off, it depends on the levels of hormone in the blood. The more hormone you have around, the stronger response you get. Second off, it depends on the affinity between hormone and receptor. Affinity just means the strength of binding between two molecules. If you have a high affinity interaction, that means you have a very strong interaction between two molecules. So if you have a high affinity interaction between hormone and receptor, it requires a lower concentration of hormone to trigger the response because the receptor is more sensitive. The receptor can detect lower concentrations. Low affinity interactions require higher concentrations of hormone. The third factor that determines how a target cell responds is the number of receptors on the target cell. The number of receptors for that particular hormone. The more receptors you have on a target cell, the stronger a response. All three of these factors may vary significantly. If you think about the regulation of the levels of hormone in the blood, regulation of hormone release, that obviously affects the level of the hormone. Some hormones will actually trigger a target cell. You may have target cells with with receptors of different affinity, so they still bind a hormone, but one receptor maybe binds it strongly, and that target cell is more sensitive, and the other receptor maybe binds it with a lower affinity, and so that target cell is less sensitive. And the number of receptors on the target cell can vary in response to exposure to hormone. Many hormones trigger a process of either sensitization or, more frequently, desensitization to the hormone. The more receptors you have on a target cell, the more sensitive that target cell is. So if you have a target cell that upregulates levels of the receptor, this target cell is going to be more sensitive. A target cell that downregulates levels of the receptor or turns it off becomes desensitized to the hormone. Sometimes the binding of the hormone itself triggers changes in receptor expression. Sometimes it's binding of a different hormone changes receptor expression. Some examples of this, insulin shows downregulation, so when insulin binds its receptor, it causes endocytosis of the receptor and the hormone, so the receptor and insulin both get broken down, and the target cell doesn't express as much of the insulin receptor. So this may play some role in type 2 diabetes. The desensitization to insulin that can lead to type 2 diabetes. It's due to this process of downregulation. Oxytocin, the oxytocin receptor upregulates in uterine muscle near the end of pregnancy, so that uterine muscle cells become much more sensitive to oxytocin, and eventually so sensitive that they go into the positive feedback cascade of labor. So upregulation tends to be a part of a positive feedback loop. Downregulation would be more of a negative feedback mechanism. Now, when a target cell does respond, there's a variety of possible ways that the target cell can change after getting stimulated by a hormone. It could show a change in membrane potential through opening and closing of ion channels, so changing the plasma membrane permeability to ions. In the anterior pituitary, this is how the hypothalamic hormones, hypothalamic releasing hormones, have their effect. Hypothalamus hormones bind to anterior pituitary cells, causing the opening of calcium channels, which triggers the anterior pituitary cells to release their hormones. Hormones can trigger protein synthesis, so that could include actually making new enzymes or regulatory molecules, could include making new transcription factors. So if a hormone turns on a particular transcription factor, then that transcription factor can go on and turn on a whole set of different proteins. The hormone could activate or deactivate an enzyme, so it could either turn an enzyme on or turn an enzyme off. It could stimulate secretion of other hormones or of non-endocrine substances. It can also stimulate cell division, like growth hormone. Growth hormone acts through mediators to stimulate division of skeletal and muscle cells. It's not necessarily an on or an off either. Hormones may change the rate of synthesis of an enzyme or another protein, so it might not necessarily turn that protein or enzyme on or off. It may just tell a cell to make more of it or make less of it. There are a variety of different types of hormones. They can be broadly divided into a couple of different categories. You have protein-based hormones or amino acid-based hormones. They may be made from short proteins like human growth hormone, or they may be derived from single amino acids, like dopamine is derived from the amino acid tyrosine. You also have the steroid-based hormones. Steroid-based hormones, like the sex hormones, testosterone and estrogen, like the corticosteroids from the adrenal cortex, these are all derived from the molecule cholesterol. Now, this distinction is important. Generally speaking, the protein or amino acid-based hormones are water-soluble. Thyroid hormone is the exception. Thyroid hormone is based on an amino acid, but it does actually cross the plasma membrane, whereas the other protein or amino acid-based hormones, being water-soluble, they can't cross the plasma membrane, because remember, only lipids cross the plasma membrane unaided. The steroids are lipid-soluble. So these guys, they can cross the plasma membrane, no problem, right? These guys can't, except for thyroid hormone. This distinction between able to cross the plasma membrane and not able to cross the plasma membrane has important implications for how hormones signal. We'll get into that in just a couple of moments. A third category of hormones are the eicosanoids. Eicosanoids they can act as hormones, but usually they act as paracrine factors. Usually they have very localized effects. There are different types of eicosanoids. They're all derived from lipids, and they have a huge variety of activities. They're involved in smooth muscle activity, which means they can control blood flow and blood pressure. They're involved in inflammation. They mediate pain responses. So mostly paracrine, but some, particularly the leukotrienes, can have hormonal effects, more widespread effects. Okay, so we know what determines a target cell. We know what can happen when a target cell responds to a hormone. now the question is, how do we get from that signal, that hormone, how do we get to the response? What happens in between hormone finding target cell and target cell actually changing? Well, it depends on what type of hormone you're talking about. We'll start with hormones that can actually cross the plasma membrane because they are a little simpler to deal with, even though there's only a few of them. So hormones that can cross the plasma membrane, like the steroid hormones, they can act directly inside the cell. All right? And this diagram shows that process. You have the steroid hormone diffusing across the plasma membrane. It still needs to bind to a receptor, only the receptor is intracellular. So that means the receptor is located inside the target cell. Steroid crosses the plasma membrane, binds to its intracellular receptor. Now, once these two are bound together, that complex can pass into the nucleus. The receptor by itself can't enter the nucleus. It's not the right shape. It doesn't fit. But once it's bound to the hormone, now it can go into the nucleus. And it can bind to particular spots on the DNA in the nucleus called hormone response elements and turn on genes next to those hormone response elements. Right? Now the genes controlled by these hormone response elements are going to get transcribed and translated into a new protein. So that's how steroid hormones trigger a response, by passing directly through the plasma membrane, binding an intracellular receptor, going into the nucleus, and turning on genes. Thyroid hormone also acts directly in the cell, but it's a bit of an odd duck. It doesn't cross by itself, but it is actively transported across the plasma membrane. Once thyroid hormone gets inside the cell, it binds receptors on mitochondria and in the nucleus. So we're still talking about intracellular receptors, but now those receptors are in the mitochondria and in the nucleus. Having thyroid hormone receptors on the mitochondria makes a lot of sense if you think back to the functions of thyroid hormone. One key function of thyroid hormone is to promote metabolism, to support normal metabolic activities, normal energy production by the cell. And since the mitochondria is the major site of energy production, it makes sense to have thyroid hormone receptors there. So, once thyroid hormone binds receptors in the mitochondria, it upregulates ATP production. Thyroid hormone bound to receptors in the nucleus bind to DNA and upregulate protein synthesis. So through both increased ATP production and changes in protein synthesis, you get the target cell response. Now let's tackle the water-soluble hormones. So water-soluble hormones cannot cross the plasma membrane. All right? They can't cross the plasma membrane. Cells don't have transport proteins to carry them across the plasma membrane. Instead, the hormone interacts with a receptor that's embedded in the plasma membrane, and the receptor triggers the response inside the cell. This process of responding inside the cell, the setting off chain reactions to change cell behavior, this is referred to as signal transduction. The hormone itself stays outside the cell. It doesn't get pulled in, but it passes on a signal inside the cell. That signal gets transduced into the cell, triggering a response. Signal transduction happens through a second messenger pathway. If you think of the hormone as the first messenger, okay, that's the first signal going to the target cell. Binds to receptor, the receptor acts through G proteins, through enzymes, and on into a second messenger. And then it's the second messenger inside the cell that either changes gene expression or causes other reactions in the cytoplasm, or both. You can think of this second messenger pathway as a bit of a relay race. You know, the hormone is carrying the signal, it passes it off to the receptor, passes it off to the G protein, to the enzyme, and then the second messenger, and then the second messenger does the rest of the work. The really nifty thing about this is that at the level of the enzyme and the second messenger, you get amplification. So you can have maybe kind of a weak hormone signal, low levels of hormone, but even though you only have a few receptors being activated and a few G proteins being activated, you can get a bunch of second messenger, a lot of second messenger activated, and you can get a strong response inside the target cell. So in a way, it's efficient because of this signal amplification. This G protein that's involved in second messenger signaling pathways is a critical step. Most hormone receptors are what are called G protein-coupled receptors, which just means that they have that G protein step. So when the hormone binds its receptor, the receptor activates a G protein inside the cell. See, this hormone receptor binding causes a shape shift, a little bit of a shimmy in the receptor that causes the shape shift in the G protein, activating the G protein. It's called a G protein because it binds GTP. GTP is guanosine triphosphate, like ATP is adenosine triphosphate. This is similar, only it's based on guanine instead of adenine. G proteins are off when they're bound to GDP. When they are turned on by a receptor, GDP is released and the G protein binds GTP. So now it's activated. And then this activated G protein can go on and alter enzyme activity in the cell. There's actually a few different types of G proteins, 3 different types of G proteins. GS is a stimulatory G protein, and it activates an enzyme called adenylate cyclase. And when the adenylate cyclase is activated, you get increase in the levels of a second messenger called cyclic AMP, CAMP. Inhibitory G protein inhibits adenylate cyclase, so with that, you get lowered levels of cyclic AMP. So it's decreasing the levels of this second messenger. GQ activates a different enzyme. It acts through the enzyme phospholipase C, and this leads to activation of a different second messenger, calcium. So through activation of phospholipase C, you get increased levels of calcium inside the cell. Now these 2 second messengers, cyclic AMP and intracellular calcium, these are the major second messengers in hormone signaling. This diagram puts together the process of cyclic AMP signaling. You have, over here, you have hormone binding receptor, activation of the G protein activating adenylate cyclase. Adenylate cyclase then converts ATP to cyclic AMP. Cyclic AMP is derived from ATP. And then cyclic AMP activates a bunch of protein kinases, enzymes that like to add phosphate groups to all sorts of cell molecules. And then the protein kinases trigger all the responses that those particular kinases activate in the target cell. So you have this step-by-step process. hormone binding receptor, receptor activating G protein activating adenylate cyclase, adenylate cyclase activating cyclic AMP activating protein kinases. And as you can see here, a number of different hormones use cyclic AMP as their second messenger. This shows you the relationship between ATP and cyclic AMP. Don't worry about memorizing the chemical structures. You just have ATP, a structure you should be familiar with its 3 phosphate groups, triphosphate. Adenylate cyclase cleaves off 2 of those phosphate groups and brings this third one around and links it back up to the adenine. That's what makes it cyclic AMP. Now there's another enzyme called phosphodiesterase that separates the phosphate group from its second bonds to the adenine, leaving you with just AMP, non-cyclic AMP. And this shuts down cyclic AMP signaling. So cyclic AMP, you can get amplification at this step and get a lot of cyclic AMP, but then it's shut back off pretty quickly. So due to phosphodiesterase activity, which is nice if you have a response that you just want a quick response and then turn it off. The other major second messenger is intracellular calcium. You get to high levels of intracellular calcium through activation of the enzyme phospholipase C. Remember, there's a G protein, GQ, not the magazine, the G protein that activates phospholipase C. Phospholipase C acts on a phospholipid that's present in the internal plasma membrane, right? So this phospholipid is PIP2. Phospholipase C cleaves PIP2 into the lipid portion, DAG, and IP3, the phosphate portion. So DAG can activate some protein kinases. IP3 activates calcium. IP3, it brings calcium into the cytoplasm both from the extracellular fluid and from the endoplasmic reticulum. So you still have hormone binding receptor, and I'm not showing that here. We have hormone binding receptor, receptor activating G protein activating phospholipase C cleaving this phospholipid, and then these 2 end products acting on the acting inside the cell. After studying your text in this video, you should be able to answer these questions. I strongly encourage you to do number 6 down here and just walk through step by step. Start with hormone binding receptor, go through what happens. I do this for both the cyclic AMP mediated signal transduction, calcium mediated signal transduction, and do it for the steroid hormones and thyroid hormone as well.